So, steps of recombinant DNA technology. So, recombinant DNA technology means it is a deliberate modification in the genetic material of an organism by direct modification of the DNA or RNA. Okay, so genetic engineering or recombinant biotechnology is also called as gene manipulation or gene cloning. And here, what we are doing is that we are deliberately modifying the genetic material of an organism by changing their nucleic acid. Nucleic acid can be RNA or DNA. So, in recombinant DNA technology, we are joining together DNA molecules from two different species, and the DNA from two different species are inserted into an host organism, and these host organisms are then again multiplied to produce certain proteins that are valued to the science, medicine, agriculture, and other industrial fields. So, coming to the steps, first step is we are isolating the target DNA and vector DNA. Then these two DNAs that we are isolated are inserted together. That is the second step. Next is cloning. Cloning means this vector DNA and the target DNA. Then it will become recombinant DNA. This recombinant DNA will be introduced into a host cell, and it is then host cell is multiplied by the process of cloning, and finally isolation of and identification of the recombinant genes for the production of proteins, drugs, etc. So coming to the first step, that is isolation of target DNA. So the DNA fragments to be cloned are called as target DNA. These target DNAs can be obtained from genomic libraries, from complementary DNA libraries, or through chemical synthesis, or even from PCR reactions. So complementary DNA clones. So what are complementary DNAs? So complementary DNAs are preferably used when the target DNA we have taken, if it is from a eukaryotic organism, then it cannot be inserted into a prokaryotic host cell. Okay, because these prokaryotic cells have different genetic organization. Here you can see that the prokaryotes DNA after transcription is converted into messenger RNA, then it is converted into protein. But in case of eukaryotes, the eukaryotes have some coding sequences called the exons and non-coding sequence called the introns. So at the time of transcription, messenger mRNAs are formed, and after that, the eukaryotic cells have the mechanism to splice or remove the non-coding sequence. Then it is converted into mRNA and then to protein chip. So if this target DNA, if it is inserted into the prokaryotic cells, the prokaryotic cells doesn't have this mechanism. That is, the splicing step is not possible in this prokaryotic cells. So for that, what we are doing is that the eukaryotic DNA is converted into mRNA in a in vitro cell culture, and this mRNA, one strand of this mRNA will be taken as the template, and into that template we are adding one of the primer, a small primer strand will be added. So that is, primers are synthetically formed or artificially synthesized small DNA strands. So one strand of mRNA of the eukaryotic DNA is taken into this, a small primer strand is added. Then we will be adding reverse transcriptase enzyme. Then what happens, according to the base pairs of this one strand of mRNA, the primer strand will start to produce a DNA strand. Okay, and this DNA, once the DNA strand started to synthesize, then DNA polymerase enzymes will be added. Then after adding DNA polymerase enzyme, the second strand will be also formed. The where here you can see a hairpin like structure. Then this hairpin structure is cleaved by some certain nucleases enzymes. Then finally you will get a double-stranded DNA. This double-stranded DNA again is called as the complementary DNA, and this DNA can be inserted into the prokaryotic cell. So next is insertion of target DNA into the suitable vector. So insertion of target DNA into a vector DNA is possible if both target DNA and vector DNA cut with the same restriction enzymes. Then only they will produce identical coherence. So if the ends are not identical, then some treatment has to be done before the ligation of target DNA into the vector DNA. So first step is that treatment with the alkaline phosphatase enzyme. Vector DNAs are treated with the alkaline phosphatase enzyme to remove the 5-phosphate group in order to prevent the vector circulation during DNA insert. Second step is the treatment with the S1 nuclease enzyme. So in the DNA, sometimes the vector DNA or the target DNA may be having protruding nucleotides. These protruding nucleotides are sometimes removed by S1 nuclease. Okay, so that is the second step. And in some other cases, we will be using linkers and adapters. Okay, linkers and adapters are used if the target DNA and vector DNA are cut with the different restriction enzymes. So if you already said that if both the target DNA and vector DNA is cut with the same restriction enzymes, then only they will be get ligated to each other. So if it is cut with the different restriction enzymes, the method we are using is that using of linkers and adapters. So linkers means they are short chemically synthesized double-stranded DNAs with blunt ends on the both side. And these linkers can be, here you can see. So the linkers will consist of chemically synthesized oligonucleotide strands with the blunt ends. So these blunt ended linkers will be attached to the both end of the target DNA, and then they are cut with the EcoR1 enzymes, and then it can be inserted into the vector DNA. In case of adapters are again double-stranded DNA with the blunt end. One end is blunt, that is this one end will be blunt and the other end will be staggered. Okay, so it can also get fitted with the target DNA and can be inserted into the vector DNA. Okay, so that means if the target DNA, for example, if you look on this, if this target DNA is cut with the if the vector DNA is already cut with the EcoR1 enzyme, but the the target DNA is cut with the BAMH1 enzymes. So this cannot be ligate. So for that, what we are doing is that either linkers or adapters can be used in order to join these two. That means in linkers means they have blunt ends. These blunt ended small strands will be fitted on the two strands of the target DNA. Then since vector DNA is cut with the EcoR1, these blunt ends will be cutted with the EcoR1, thereby these target DNA can get inside the vector DNA. Adapter molecules means these adapter molecules, one side will be blunt, the other side will be staggered. So these staggered ends can be fitted inside the staggered ends of this vector. So that's all about linkers. Then sometimes some other process method can be also done in order to link the vector DNA and target DNA. The fourth process is that homopolymer sequences or tailing. For example, if both the target DNA and the vector DNA has bundles, so we already known as is known that if they have the staggered ends only, they can be ligated. So if the target DNA and blend DNA vector DNA have blend ends, then it cannot be ligated. For that purpose, we can do homopolymer sequencing. That means here you can see this vector DNA, the both sides. That means especially the 3' position, you can insert a small polymer of adenine, and the next strand also small polymer of adenine is inserted. And in case of DNA fragment, as adenine can base pair with the thymine, the DNA fragment one end can be add a thymine polymer, the next in can be add another thymine polymer. Then while ligating, what happens? Simply mixing itself, these two will get ligated to form a recombinant DNA.